All right, I guess we'll start. So, hello, everyone. This is Category 5 workshop. All these events will be biology-based. So we have anatomy and physiology, designer genes, disease detectives, protein modeling, and ornithology. Thank you all for coming. So, I guess we'll just start. So, once again, we have the events listed here. They're all going to be biology-based. So, an understanding of cells, DNA and their interactions will be a common theme in all these events. So first up for anatomy and physiology. So this is an event that covers the human body systems and their functions. You're in teams of two, and it consists of a written test only, which has multiple choice questions mostly, with also a free response. There's a lot of labeling in this event because you have to label the muscles and bones and layers of the skin, and explaining for some of the answers. You're able to bring one cheat sheet. It has to be only one printer-sized sheet of paper, but it can be front and back as well. You can laminate it as well. We recommend you put diagrams on that, but I'll cover that more in a bit. And each year, the systems rotate, right? So for this year, since they're the same as last year, the systems are the integumentary system, the skeletal system, and the muscular system. So what do you need to know for the integumentary system? So obviously, most of the questions are based off of layers of skin. So this is knowing your epidermis, dermis, and hypodermis. So more specifically, you have to know your layers of the epidermis, like your stratum basale, stratum spinosum, stratum granulosum, and corneum. I have a diagram listed there. And to know their individual features and qualities, right? Because most of the times in the questions, they'll ask you about like, what are the cells in the stratum basale like? You need to describe them. And also understanding and knowing what they look like also helps a lot. And then we also have the layers of dermis, which is we think of features and qualities. So this is like the reticular and papillary layer. And to know like what type of connective tissue there are, which is pretty important considering they have different structures. And another important feature is knowing what type of skin organs or accessory organs are in there, such as hair follicles and blood vessels. But for the sake of time, for the tryouts, we won't be covering those. Just know the difference between the papillary layer and the reticular layer. And then the different cell types are also very important. This is knowing your melanocytes, keratinocytes, and the specialized cells that may appear in the skin. And then knowing the functions as well, such as protection, both physical and microbial, vitamin D synthesis, and heat is also very important. There's also more functions which are all covered in the resource packet that we sent out. And then disorders, this covers cancers, burns, and irritations. So attached resource packet is a textbook that I sent, or I cut down the pages of the textbook to send you, right? So please look through those to find the skin disorders, because it's important that you're able to research these things on your own. And we'll be testing that. And so for the skeletal system, most of it is based off the bone structure. So this is knowing like visible and cross-sectional. So knowing your Haversian system for the cross-section, and knowing your diaphysis and epiphysis when you look at the visible structure, right? And also differentiating spongy bone and hard bone, and knowing what type of marrow are inside each bone, and the types of bones as well. And then for bone cells, knowing what each bone cell does and how they contribute to the structure and growth of the bones. It's also very important. So for most of the tests, right, they'll ask you to label a few bones of the axial, which is like your spine and cranium, and appendicular, which is like your limbs and other parts of your body. So they'll ask you to label these bones, right? However, for the sake of the tryouts, we will be testing over these. However, just know that when you actually test over the anatomy and physiology, make sure you have something on your cheat sheet so you can check over this with that, because they're hard to memorize. We have a lot of bones, of course. So just remember to keep that in mind. Also knowing the functions of the skeletal system and types of cartilage and their uses, which are all covered in the packets once again, and our skeletal disorders, which is including fractures and degenerative diseases such as rickets or osteoporosis. And then for the muscular system, understanding the muscular hierarchy. So what I mean by this is understanding like the fascicles, fibers, and myofibrils. Like the order of which they go down, and the connective tissue surrounding them, because all the times they'll be asked label questions about these. And then we have cellular and gross anatomy. So what this means essentially is that this is one of those complex parts of the muscular system. It'll ask you to explain how the muscles move at the cellular level. So at the bottom right, you can see that I put a picture of a sarcomere. So basically how this works is those thick filaments in the center, also called myosin filaments, they have little filaments that attach to actin filaments at the top. And so when you contract your muscles, they'll inch slowly and contract the sarcomere. And so sarcomeres, we have like millions upon billions of sarcomeres in our body. So each of these contract once, and then when all of these contract at the same time, you have a muscle contraction, of course. And so when they relax, the filaments connecting to thick and thin filaments, they relax, right? And they release. And so they expand out once again. So that's the cellular part. And the gross anatomy, understanding like antagonistic pairs, how our muscles work in pairs to let them go back and forth. And then in your muscle types, which are cardiac, smooth, and skeletal, a lot of times they'll ask you to differentiate between them. So understanding which are striated and which does what type of contraction. And then our functions of muscle, which are movement and heat and a few others, and our muscular disorders. However, for tryouts, I won't be testing over this because I think it's more important as of now to understand sarcomere contraction, which I've labeled in the textbook pages once again, and the rest are covered in the packet. So a few tips. A lot of the information in anatomy and physiology can be overwhelming, right? Because it's a large topic, there's a lot of information you have to cover. So what I'd recommend is going through first the Khan Academy. The Khan Academy has 3 playlists, right? One for skeletal, integumentary, and muscular. Just watch over those, maybe take a few notes. Those are really helpful in understanding some of the harder concepts. And then I think honestly the best way to understand anatomy and physiology is to just read the textbook because they cover most things, right? They go in depth, they have good images are also really important, right? Because understanding what they look like is very important because on the test, they'll usually include like a diagram or two, which you have to then label anyways. So understanding what they look like, understanding just what they look like, yeah. Understanding what they look like and what they do is very important. That's like 90% of the test. And then ties, right? Ties. So ties are broken based upon 1 or 2 written responses. So usually it's one of the harder topics. They'll ask you a question or they'll ask the label steps of a process. You have to answer that, and the team that is most accurate or provides the most amount of information wins the tiebreaker. So those are really important in deciding where you could place if you're already doing well in other sections. And then cheat sheet wise, so generally for your cheat sheet, you want to have mostly diagrams because anatomy and physiology is mostly made up of diagrams, right? However, it's more valuable and more resourceful of you to memorize information rather than spending hours upon hours of memorizing diagrams. Memorize the diseases, memorize the letters, memorize the functions, but maybe keep like a skeletal diagram or a muscular diagram because it helps you save so much time on labeling, and it's really easy for you to just check your answers with. And then the question for this section to make sure you were paying attention is, what are the 3 body systems for the 2020 to 2021 season? So test this to Madia privately.
And Madia, let us know when you got all the responses on most of them.
Oh, Tracy, you want to add anything?
Well, I think you pretty much covered everything. Can you hear me?
Yeah.
Okay. So, yeah, it's just, I remember when we did it, it's a lot of diagrams and labeling. So for the actual competition day, for cheat sheets, don't focus on stacking your cheat sheets with like a lot of information and like having a size 8 font. Just mainly fill it up with diagrams and then try memorizing the other information. But for tryouts testing, yeah, the Khan Academy videos help a lot.
Oh, I wanted to add on something. You guys can just text me privately through the chat section. You don't have to like text me through Discord. So yeah.
You can do that, by the way, on the chat on the bottom right where it says to and then colon. You can like click on everyone and it'll give you options, and then click on Madi or Raman, and then you can privately text her that way.
Oh yeah, if you have any like questions about the event, you can ask them now. I can answer them.
I think I have all the responses. You can move on.
All right, cool.
All right, so designer genes. So what is designer genes and what does it cover? So designer genes is a study event, just like anatomy and physiology, and it covers, as stated by the rulebook, genetics, molecular genetics, and biotechnology, meaning that designer genes basically covers most of, if not all of, biology. And one thing I want to point out before going to the event details is that if you've taken pre-P biology, or if you've taken AP bio, or you're planning on taking biology, or you're planning on self-studying biology, this event is for you. And like this is solely based on bio. Like everything you learn about biology in your 9th grade class is the foundation of this event. Now, of course, you have to keep in mind that Science Olympiad is more advanced. The tests you get in school are going to be nothing like the tests you get in Science Olympiad, but the basics are there at school. All you have to do is self-study them, review them, and then research on your own the more advanced level of those topics and be able to apply them. But generally, if you have that drive to self-study bio, go for it. And one thing that's really cool about biology in relation to chemistry and physics is that it's a lot easier to self-study, because for chemistry and for physics, you have to know a lot more math. You need to you have a lot of concepts to know. Bio, it's more like memorize some terms, understand the processes. So if you read through a bio textbook, you'll get most of the points that you need to know. And you just need to, there are a lot of resources online for you to use for designer genes. It's been an event for Science Olympiad for a really long time. So if bio is like where you want to be, this is a very recommended event for you. So now on to the event details. So on the designer genes test, actually, wait, first off, designer genes is a study event. So the only thing you have to do for designer genes is take a test. So you don't have to build anything, you don't have to do anything. Just go to a classroom, take a test, do well. So the questions you can expect on the test are multiple choice and free response. You can expect both types of these questions. And just like anatomy, you can also have a cheat sheet front and back. And again, we do recommend diagrams, except I'll go into more details in the next few slides about that. And then finally, get a Campbell biology textbook if you want to ace this event. So, okay, start off, what is a Campbell biology textbook? So the Campbell biology textbook is a textbook written by this biology genius named Campbell, who's written this textbook for people all over the world to study and know biology. It's mainly for high schoolers. And this book has been used for people who want to study and ace the biology Olympiad, if any of you guys have heard of that. And it's also used by many designer genes participants. I actually bought the textbook. I bought this one. It's written by Campbell as well as some other biologists. But if you want to do well in this event, you need to get some kind of textbook like that. You need to read through it throughout the year. It's really not that hard if you just spend like 10 or 20 minutes a day reading it. But if you are able to read through this Campbell biology textbook, and if you're able to understand everything, you're on your way for a perfect score in this test. It's really very easy to self-study if you just put in the time and effort for it. So please keep this in mind if you want to pursue in designer genes after the tryouts test. This is something you want to do. Remember your Campbell biology book. Pre-order it. It's I don't think it's that expensive. You can find it online, whatever the latest edition is, but it's very recommended. Slide. So here I've listed, or actually I screenshotted the table that was provided on the rulebook that covers all the topics you need to know for designer genes. Of course, for tryouts, you don't need to know this. This is not for tryouts, but I just wanted to show you guys an overview of what designer genes looks like and what topics it covers. So if you just briefly skim through the table, you'll see that a lot of the topics here you have covered in pre-P bio if you've taken that already. For example, mitosis, meiosis, or codominance, incomplete dominance, the DNA structure. All these things we've learned, and some things on here you might have not learned, so you can just you can research that on your own. But what I'm trying to say is that, again, as long as you have a bio background, this is a good event for you, because all you have to do is research it and apply it. And if you look at the resource packet already, you'll note that a lot of the notes, actually I think all the notes I gave you for designer genes are from my bio packet from 9th grade. So again, this proves my point. It's very bio heavy. So again, just study it on your own. This is a good event if you have interest in biology. Okay, so now that I showed you what you need to know for designer genes in general, if you want to do it this year, let's take a look at what you need to know for tryouts. Because if you look at the table, that's a lot of stuff to know. There's no way you can read that textbook in 4 weeks and study all the other events, right? Biology is a huge course. So I broke it down for you, and here are the bullet points I included in the resource packet that covers the topics you need to know for tryouts. So I'm just going to run through this real quick. So you need to know mitosis, meiosis, and gamete formation, pedigree construction, and analysis, like the ones, like those circles and triangles, that diagram. If you guys remember from biology, it shows you your ancestry. You need to know multiple alleles and sex-linked traits. I specifically included that you need to know examples of multiple alleles or examples of sex-linked traits, because a lot of designer genes tests will actually ask you for an example. So, for example, color blindness would be an example of a sex-linked trait. DNA structure and mechanism of replication. So you need to know what makes up DNA, how does it replicate, which goes on to transcription and translation, which involves the proteins related to DNA. And then finally, you need to know how to make a cheat sheet. I didn't include this in the resource packet, but you do need to know this for all the events in this category other than ornithology. So again, I'll get to the details of that in a minute. But again, I've included my bio notes in there. So if you read through my bio notes in the resource packet, you'll be able to answer the level 1 and 2 questions on the tryouts test. But when it comes to the level 3 questions, you're not going to be able to answer them unless you know how to apply the knowledge and unless you do some research on your own. Like, just search up the topic designer genes practice test and you'll find loads of stuff. That's how you can test you on that. And if you want to do designer genes, we expect you to at least be able to do the level 1 and 2 questions and then try the level 3 ones so that we know your potential for the advanced bio. Okay, so Tony already explained a lot of the details for cheat sheet, but I'm going to elaborate a bit. So I provided 2 examples of some cheat sheets that I see at tournaments. The one on the left, believe it or not, is actually widely used among a lot of people for some reason. Like, every time I go into the designer genes room or for anatomy, there's always people with like papers with size, I don't know, 2 font, and there's so much text on there, and they have like little tiny highlights, and then there's like 1 or 2 diagrams on the back, and then whenever it comes to the test, they're like flipping through, like holding the paper like this, and one partner is going here and the other partner is going there, and they're trying to look through all the text. That's not the cheat sheet you want. Just like Tony said, there's no need for you to make a cheat sheet that heavy. And if you really think there's a need to do that, there isn't. Trust me. First of all, you will not have the time to go through all that text. You simply don't. You have 50 minutes to complete this designer genes test. It's usually pretty complicated. You can't go through it all. And then let's say you do have the time. You're not going to, like, when your adrenaline is kicking, you're not going to you don't have the patience to go through that whole cheat sheet and figure it out. You want answers fast. That's your goal, right? So that's the worst kind of cheat sheet. We don't recommend it. From the graduates who have graduated from Science Olympiad, they've also told us not to do that. On the other hand, on the right, this is actually an example of a cheat sheet Taha and I used this year. for designer genes at regionals. And it was pretty helpful because, as you can see on here, there are a lot of diagrams. And these diagrams not only show us the different parts of the DNA or whatnot, but it also explains most of the topics that you need to know. So if you need a quick reference, there's your diagram. And it's not like we're telling you not to put any text. We did put some text on the bottom left, if you can see here on a cheat sheet. And I think for anatomy, Tony mentioned to like put some stuff on there because it's hard to memorize for a certain topic. So you can do that stuff, but we don't recommend putting all of the information like that on a cheat sheet like it is on the left. So for tryouts, what you're going to need to know for cheat sheets, how you are going to make it and what is the most efficient way to do so. And I would really recommend for you to photograph this slide into your memory so that you understand what we're talking about when it comes to the test and for in general and future reference. Okay, so next up, I have some examples of 3 types of questions that are usually seen in designer genes. A lot of this also applies to anatomy and physiology, by the way. So starting with labeling questions. So Tony did mention this as well, labeling questions, how it appears so much in anatomy. And it's true, a lot of it appears both in anatomy and designer genes. For anatomy, they usually appear at the beginning of the test. Designer genes, it's usually scattered. I've never seen it at the beginning. I don't think I have, at least. But basically, they give you a diagram of something for designer genes like DNA or protein, whatnot, and then they'll ask you to label it, just like as you see here, number 37 to 45, you have to label them. And at first, when you look at this, you're confused already. There's no way you probably can memorize all of this unless, you're a bio genius and you really can't memorize it. There's a lot of diagrams out there to know, so I kind of doubt it you would know every detail, especially the bonuses they give in here. But if you look at that cheat sheet I had on the previous slide, I actually had this exact same diagram on the cheat sheet in the top right corner. So because this diagram is on my cheat sheet, it's so easy for me to fill this out. Now all I have to do is look at the cheat sheet, copy the answers, and I'm done. And I probably will get a perfect score if my diagram on the cheat sheet is right. So that's why we say put diagrams on a cheat sheet, and especially for anatomy, they repeat. I don't know why, but they usually copy and paste a lot of the diagrams that are on Google, like one of the first results. It's happened so many times. So again, cheat sheet diagrams will help you in label questions, and they'll be a breeze. And now we have free response questions. So for designer genes and for really any study event, you will expect to see free response questions. And here I've given 3 example problems of such free response questions that usually appear actually in designer genes. So starting with number 1, they've given us a situation, they've given like the genotypes of a man and a woman, their children, and the question is asking what's the genotype of the woman, and it's asking what kind of gametes can she produce. Okay, so number 2 is giving more information on number 1, and then it's asking what kind of gametes can the man produce. And then number 3 is asking you to fill in a Punnett square. So these type of free response questions are really they always appear. These always appear. I've never seen a test that doesn't ask you for a Punnett square. I've never seen a test that doesn't give you a situation and ask you for the genotype or gamete or phenotype. So the reason why I'm telling you this is because for tryouts, please expect these type of problems. I don't think I've listed Punnett square as one of the topics we're testing you on, but I do believe we're testing on pedigree construction and analysis, and those tend to be one of the harder questions because they're not usually multiple choice, they're usually free response. So be prepared for that. Be prepared to have a picture of pedigree construction and then an answer about maybe one of the people on the pedigree. So again, I provided notes so that you know how to read the pedigree. Again, there are advanced questions though, so maybe do some practice problems on your own. And then again, be prepared for free response questions. So I remember sometimes designer genes tests don't even have any multiple choice questions. Sometimes they have only free response questions. Or for protein modeling at Rice, there were no multiple choice questions, and the highest score was a 25%. So they like to do free response questions to trip you up. So be prepared for that. And then finally, multiple choice questions. So these are usually on the easier side. I think this is the easiest type of question other than labeling if you have a good cheat sheet. Here I've provided two different types of problems you can find. These are very basic problems. I would say they're like level 1 or 2. I didn't give you the advanced ones. I just want to show you what topics are covered. So just taking a look real quick at the first one, number 32, they're giving you a DNA strand and they're asking you for the complement DNA. So here you would just need to apply your knowledge on complementary strands, and you would find the answers A, yes, because the 58 and 38 are switched. A is matching with T, C is matching with G. For number 33, the second one, the portion of the DNA molecule that can vary is its. So for this question, you would need to know the structure of the DNA and you would need to apply it. So this is more of an application question. Because as you can see from all these answer choices, these are all parts of the DNA, but it's asking which one is different. So if you know anything about DNA, you would know that there's a sugar group, there's a phosphate group, and a bases. So A is out, C is out, D is out, E is out, because bases is the only one that changes. So that would be an example of an easy application question you can see on trials too. I think that's it. Before you answer this question, I think that Taha's here. So Taha's been my partner for a while, and he's also a biology genius. He's known for that. So if you would like to add anything, any study tips for the people who may want to do this event this year, Taha?
Yeah, sorry, my video isn't working, but anyways, I think what's something that you really should practice is like pedigree analysis and genetics questions, especially ones that deal with probability. So I feel like everything else you can kind of just study and like learn what it is, but those ones specifically, like you can't get really good at answering those questions unless you like do a lot of them. So I think it's important to do that. And as for like textbook resources, I think Campbell Biology is just overall like the best bio book you can use as a high schooler. So just you can go, they have like one of the units is just genetics. So like just reading through that and then seeing where you need to fill in gaps, I think would be a good idea.
Okay, thanks, Taha. Yeah, so a lot of practice and then the Campbell Biology book. That's really good. Okay, so we're gonna give you a minute to answer this question. Also, if you have any questions for designer genes in general, you can let me or Taha know. We can answer them. And thanks, Taha, for coming today.
I think we can move on now. I have most of the answers.
Okay, cool.
Okay, so next we have disease detectives. So this event will be a written exam like the other two, and then it'll be about epidemiology and identifying / investigating it in different related scenarios and populations. So for some event details about disease detectives, it will be both multiple choice and free response like all the other written exams. You'll be allowed the cheat sheet, and just like Tony and Angelica said earlier, you want to fill it up with like diagrams and not like pack it up with a lot of information. Only like allow like the long list. So like for cheat sheets for disease detectives, there's a lot of formulas you should know. So filling it up with the different formulas like prevalence, incidence rate, relative risk is really good. And also a lot of the questions in disease detectives in the past years ask about history of epidemiology and populations. So obviously you're not going to remember like every single like case of every single case of like pandemic that's happened over the past like centuries. So it'll be good to list that out in your cheat sheet, and also the 13 steps for like observation and that stuff. For disease detectives, also, the three main topics that will be covered this year and all previous years is background and surveillance, outbreak investigation, and patterns, controls, and preventions. And you can find more information about this in the Cellular Rulebook and also in our resource packet that we provided you with. And for this event, because there's a lot of formulas and calculations, you will also be allowed to use calculators. So some need to know for the tryout test, make sure you understand and know what epidemiology is. So epidemiology is the study of diseases and like analysis of diseases and like populations. And also there are also know the key terms that relate to epidemiology. We have provided a glossary in the resource packet of all the terms that are important that show up in the test. But obviously we won't test you out on like all of them because that's a lot of information. We listed out some important ones on the side in parentheses that you should know, like what hypothesis is, what a pandemic is, etc., like the main ones. Also, as I mentioned earlier with calculators, make sure you understand the formulas and when to use them. In the disease detectives test, a lot of it is filled with graphs and tables and data, and it'll ask you to calculate like what's the percentage of exposed or unexposed, or what's the prevalence or the relative risk ratio. So make sure you understand these formulas and are able to use them and know when to use them. And again, like I said earlier, there's a lot of graphs and tables and data sprinkled around because you'll be analyzing like diseases in different populations. So the two main types of graphs and tables that are mainly used are epicurves and 2x2 tables. So epicurves are a type of histogram, and it basically just shows like the curve of those who have experienced the disease over a period of time. And then a 2x2 table is, like I said, it's 4 boxes, and it basically has exposed, unexposed, and then if they have the disease or they don't have the disease. And usually for 2x2 tables, they ask you to calculate the odds ratio and the relative risk. So be able to use the formulas in these tables and datas, and also read the information provided above, because there might be some errors or biases which you will have to identify, which again are listed in our resource packets. Next, a huge chunk of disease detectives is diseases. So make sure you understand like what diseases are and like how they're transmitted, like the modes of transmission, like waterborne, airborne, vector-borne, foodborne, and that stuff, and also understand how it is controlled. So in our resources packet, we have a diagram, and it shows the steps of control. So the first step would be control, and then elimination, eradication, and extinction. Also, know how to prevent diseases. We also have that listed in our resource package. And then the chain of infection. So the chain of infection basically is how the infectious agent moves around and infects the host of the body. So it needs a place to grow, and then it exits, and then it needs a way to be transmitted into the body of the host. So that's basically what you need to know for diseases. And then you would also need to know foodborne illnesses. So from my experience in past years, foodborne illnesses isn't a huge chunk as many as it mainly is about like studying cases and like different scenarios. So mainly they use foodborne illnesses as a tiebreaker round. So they ask you to like identify like which foodborne illnesses went with this year, or also a common question is like steps to safely prepare food to prevent foodborne illnesses. So you need to know that, which is we've learned before in like science classes, and also you learn in health classes. So it's good to read up on that. And in our resource packets, we actually have a CDC and FDA chart of the different types of foodborne illnesses organisms. So it's also good to read through that just to get a common knowledge. and then also know the acronym FAT-TOM. So FAT-TOM is an acronym for the 6 favorable conditions that foodborne illnesses grow in. So it stands for food, acidity, time, temperature, oxygen, and moisture. So it is really important that you know this because it will be asked later on in our tryouts test and also in the disease detectives actual day competition. And then last is the last 2 categories that you'll be tested on in disease detectives, which is surveillance and then outbreak investigation. So make sure you understand what surveillance is, how it's used, and what its purpose is, and also know the 4 different types of surveillances, which you can find again in our resource packets. And then for outbreak investigation, it mainly talks about clinical versus public investigations. So make sure you understand what the focus of clinical and public is and what it's centered around. So clinical focuses on individuals and it tests and diagnoses, and then public focuses on populations and it controls and prevents. And then for outbreak investigations, also know the different steps. So for outbreak investigation, there are 13 total steps for it. And obviously, we don't expect you to memorize all of it, but just read through and understand, and at least memorize step 1, 2, 4, and 10. So step number 1 talks about identifying it, and then step 2 talks about is it considered an outbreak, and then step 4 is about how to construct a case. So it's really important that you know how to construct a case, so read up on that. And also step 10 is about the Bradford-Hill criteria. So make sure you know that the Bradford-Hill criteria is part of step 10, as they also ask you to use Bradford-Hill criteria to identify this type of case. And then also know the different types of studies for outbreak investigation. You have both observational and experimental, and then know about the history. As I mentioned earlier, they'll ask like what happened in 1500 BC, or like when did the HIV outbreak happen, or when did the SARS happen. So make sure you read up on that, which we have listed in our resource packet again, and we have highlighted like key terms that you should know too. So the next slide we have is a couple of pictures. So as you see in the top left-hand corner is an example of a 2x2 table, and you can see it has the odds ratio formula listed below, and you just plug in where A, B, C, and D are, and you solve. So make sure you understand what this formula is and how you can use it. And it's also very a good tip is to include this in your cheat sheet for the competition day, as you want to save time memorizing other materials instead of formulas, and you just stick that on your cheat sheet. And then below that is an example of an epicurve. As you can see, it looks very similar to a histogram, and it basically just shows how many people were infected over a period of time. And then the top right corner is a picture of the acronym FAT-TOM, which you should know is the 6 favorable conditions for foodborne illnesses to grow. And then below that is the chain of infection, which starts out with infectious agent, and then it grows somewhere, and then it exits and is transmitted by type of mode of transmission, and then it enters a host and it starts to infect you. So make sure you know these pictures, and then we have a little question for you guys to answer. So make sure you guys text it privately to Madia. So the question is, what is the acronym for the 6 favorable conditions for foodborne illness growth? And then Tony, do you want to add anything to disease detectives?
Yeah, I'll just say that at first, when I tried this event, I thought it'd just be random questions about disease, but most of the questions are based off a narrative. So they'll give you a situation, and you have to apply your knowledge there. So it's not as straightforward as you might think.
Yeah, in general for Science Olympiad, and especially for this category, it's not enough to just identify stuff. You've got to apply it. So it's like Tony said, maybe at first he thought it was going to be just about diseases, but you have to actually apply it to some situations. So the same goes for all the events. So as you study for it, as you study for this category and for other categories, know that. Just keep that in mind because it'll help you study efficiently.
Okay, I think we can move on now.
All right.
Okay, so next is protein modeling. So protein modeling is a 3-portion event. You will build a model of a specific protein, and this year it is cytidine deaminase. And then you also explore a new protein on a computer and take a test. So this year they updated the event. So instead of building another model at the event, you'll just be exploring a new protein that they give you on a computer there. So you won't have to worry about constructing another protein. So some event details, as I stated earlier, is that it's 3 portions. You have a pre-build, which will be of the protein cytidine deaminase, and then you explore JMOL. So JMOL is a computer program, which I'll talk about more later, and you basically explore it on the day of the competition, and you'll answer some questions, and then you will also take a written exam about the protein that you build, or about the CRISPR-Cas9 tool. For this event, unlike others, it is 3 partners. So you is a team of 3. So instead of usually 2, you have 3 people. And then again, you're allowed a cheat sheet for the written exam. So make sure you follow what Tony and Angelica said earlier about cheat sheets and understand how to create an efficient one. Also, don't worry about bringing a computer to the protein modeling competition day. They'll provide you with one there, and then to win, you just want to be accurate. Make sure your model for your protein is accurate. It follows the correct scale thing that the rulebook tells you, like 2 centimeters apart per amino acid. And then for the computer and then the written test, just make sure you answer the questions thoroughly and correctly. Just add a lot of details if you can, or just as much information that you know about it. So some need to know for the tryout, just know what needs to make up the model. So like the carbon backbone and like the protein folding, have like an idea of a design that you would create for the protein that we're using this year and be able to describe it. And also, like for other build events, have a list of materials and supplies that you could use. So something commonly used is mini tubers, or something like quick twist, or something that's bendable that allows you to construct your model. And also, a thing to note is that you're not allowed to use 3D structures for your actual protein, but your relevant features can have 3D printing on them. So next is JMOL. So as I stated earlier, you'll be using JMOL for your pre-build and also for the computer exploration on the day of the competition. So basically, JMOL is a computer program that will allow you to visualize your protein structure before you build it, and you'll basically have to like color code some portions of it and that stuff. And we have a list of instructions in our resource packet, which leads you to the Cellular Wiki page, and it basically explains to you how to download it, how to get the data, and also how the different modules work. So during this time while you prepare for the tryouts, feel free to download JMOL and explore and mess around, because we want to make sure you have a familiar understanding of JMOL as you will be tested on the competition day too. So just download it on your computer or laptop and just explore around. And then, as I mentioned earlier, for the protein modeling build, you also have to include relevant features onto your model. So examples of relevant features would be like amino acid side chains or other molecules that are related to it. So as you are studying up on this event, think up of some features that you can add to the protein and make sure that you're able to follow the index card format of those. So the index card format is something you have to have with your protein modeling on the day that you submit it. So what it has to include is what is being displayed of your relevant feature, how is it displayed, and why is it important. So make sure you have a good understanding of the relevant features you choose as well. And then for the written exam, you will need to know specific topics, which you can find more on the Cellular Rulebook. And also, I have linked a few resources that you can read up on. So what you would need to know is what protein folding is, CRISPR-Cas9, chemical principles, and how it stabilizes. So protein folding is basically it takes its functional shape to perform its correct function. So if you've ever taken biology or will be taking biology, one of the main things that they say is that structure determines function. So make sure you understand what protein folding is, and then CRISPR-Cas9 is basically a technological tool that allows you to edit a genome. So you can alter or delete or add like specific DNA sequences. So make sure you understand how CRISPR-Cas9 works and be able to apply it to your protein model, as this event will be testing how you can edit plant and animal genomes. And then also make sure you know what the protein this year is. So the protein this year is cytidine deaminase. So make sure you read up on that, understand how it works, and then how it relates to CRISPR-Cas9. So on the next slide, we have a few pictures. So on the right top is a protein example of the cytidine deaminase, and then below is a few examples of past protein modeling examples. As you can see on the left side is what it looks like on the computer, and then how they recreated it in real life. And then on the left side is a model of Angelica's protein that she did this past year. So Angelica, do you want to explain more on protein modeling?
Yeah, so for the build portion, so there's a website they give you on the rule book for you to refer to for the build portion and the JML exploration. For the build portion, you need to make sure you're as accurate as possible, just as Tracy said. So you can see on all these models, like the way the spirals are made, or like the direction of the spiral matters as well. And like Tracy said again, the shape determines the function. So if you get even a little curve wrong on the protein, it can make a lot of impact, and they will score you on how accurate you are. And you can just see on the model like some lines I've written. Those are like the requirements they need you to have in the model. So, or like the caps, the color caps. They need to read the rule book to make sure you know or have a general understanding of what you need to include in the model. Because we might test you on that, because you can't necessarily draw this out, but we will test you on what you need to know or what you need to have on the build. For the computer exploration, they do have a PDF of a list of commands that are useful for you to know. So you need to be familiar with at least the basic commands. We were going to test you on that. Because we can't really require you to have the JML open when we're taking the test, so we will test you on some of those commands. So you might want to look into that. There are also quizzes probably online for people who made it this year. You can find that. And if you have any questions, you can DM me about it, because it's kind of hard, the exploration computer thing. It can be confusing at first. Also, in order to build a model, you have to use the JML thing. That's a big thing, because at first some people don't know how to build it. You don't have to search it up on Google Images. You have to actually go to the JML exploration and figure out the commands to find the different parts. And then finally, for the test portion, I would say the test is really advanced. So every protein model test I've taken, they've asked really advanced stuff, like advanced molecular biology. It's super detailed. I don't think it covers a lot of stuff in 9th grade bio. I think it's more in AP bio and self-studying. And like I said, I think I said this earlier, but the highest score at Rice was a 25. So again, out of 100. So it's really hard. So if you want to do something biology related, you can do designer genes or protein modeling, or of course any of the other events. But those are the bio heavy ones. But protein modeling is more on the advanced side. So just keep that in mind. But it is a cool event, so I will recommend it. Also, if you're taking chemistry and you made that molecule this year, it's kind of like that before protein. So that's all I have to say.
Okay, so next we have a question. So again, make sure you text it privately to Maria. So the question will be, what is the name of the protein you will be building a model of this year?
I think you should continue. Nobody else is responding, so go ahead.
All right, so ornithology. Ornithology is basically an event where you would focus on memorizing a lot of info and then working with a partner. This event tests you on basically identification of North American birds by providing you a picture of the bird or some sort of like skeletal structures or part of the bird's picture, like part of the bird's like body or something as the picture shows, and recordings of songs. So basically, ornithology is an event like with a team up to 2, and you could have a binder of 2 inches or smaller. And for the binder, you would have to include about 160 birds. So it is really important that you understand or you just plan out how you would organize it before you try out for the event. The binder could contain any info that you might need, and along with the binder, you could also use a commercially produced field guide. And the field guide is basically like an already made binder that could help you with this event. This event is like structured like a slideshow presentation or a series of stations that we kind of do at school sometimes. So for a slideshow presentation, they would normally have questions on the top of the slide, and then they would have like a huge picture for you to see. I have a picture on the next slide, and I'll show you that later. And then stations would also, so like slideshows and stations both will be timed. So you have like a minute to answer 3 questions. And for stations, it would be just similar with what we do at school. The supervisor that's in charge of this event would like time it for you, and then you just have to listen to her instructions. And next slide, please. All right, what do you need to know for tryouts? So first, you would have to know how you could effectively construct a binder that is actually helpful. For example, if you construct a binder that has a page of info for 1 bird, and you do that for 160 birds, and on event day, you would only have 1 minute to answer 3 questions like the picture shows. How would you manage to flip through, or like how would you know where to go to find the answers? And if you, for example, you would need at least 30 seconds to look up or to find the page that you need. And it is really crucial that you understand how to, like, what are the ways you could organize your binder, like having labels, organizing the birds in alphabetical order or anything like that. And although like just combining like 160 pages in 1 binder is like the simplest way to like construct it, but it might not be too helpful in any way. And as you can see on the bottom, we chose 7 birds for you to know for tryouts, and we understand that it is impossible for you to study or research on 160 birds. So that's why we shortened it to 7. And for those 7 questions, for those 7 birds, we'll ask you questions on their common and scientific names, their order, family, or genus. For those like identification questions, just like identifying or like memorizing like straight facts, those are going to be like level 1 question. And for more of advanced questions, we'll ask you to find common characteristics between those birds. For example, like what are the following birds that are in the same order or family? And a more advanced question could also be identifying the bird by listening to a recording of songs. And yeah, and then you would also have to know the general knowledge of anatomy as well. For example, if we give you like a skeletal structure of a bird, and then we might ask you to label certain part, and for more of an advanced question, we might ask you to like explain how that structure functions and something like that. And that means you really need to focus not only on like identifying and memorizing straight facts, but also like studying the internal structure of a bird as well. And yeah, for distribution and occurrence, that's basically asking you, is this bird common or rare in Texas? Like number 3 on the picture, should you expect to observe this bird year-round in Oklahoma? Questions like that. Those will be level 2 or 3 questions as well. Next slide, please. Okay, pictures. The first picture on the left is what I meant by studying the structure or the internal parts of the bird. 31 and 32 would be level 1 questions that would ask you about just like identifying the structure. And then 33, what is the function of the structure, would be a level 3 question where you need to explain it. And yeah, and for a field guide, I would really recommend the Sibley birds, which was what I used for my event last year. And it was really helpful because one partner could mainly be in charge of the binder, and then somebody else could, use the field guide to find information as well. And like I said, it's just a made binder there already for you, so there's no harm of using it. And the last picture on the right is an example of the binder that Kathy and I made last year, and it's in alphabetical order. And then we also had like the general knowledge of ornithology in the front, as well as like table of contents with scientific names, and another table of content with only picture and the common name of the bird. So for questions like identifying the picture, identifying this bird, you would it's easier to go to the table of content and look for that picture or a similar one, and then look for the common name first, and then go through the alphabetical, like the labels, and find where the information is. And I think that's pretty much it. Next slide is a question for you. What is the size of the binder you could bring? Oh, by the way, all the study resources or information for this event could be found in the Cornell Lab of Ornithology, which and other links that I included in the resource packet.
Okay, we can go on now.
All right, so that is the conclusion to our workshop. If you have any questions, now is the time to ask them. All right, so if not, have a nice day, and thank you for coming.